All right, so I will start discussing DNA replication in this recording, this part 2 of chapter 16. Like I told you as I end the part 1 when I introduced this chapter, I told you that DNA replication cannot begin by making DNA. It begins by making RNA. You don't make DNA by making DNA. You make DNA by first making RNA. So how does that happen? So whenever you want to make a DNA sequence, you have to start by making RNA first. This is the reason. The enzyme that makes DNA is called DNA polymerase. This enzyme is a big enzyme. Think about it that way. They are not able to start by themselves. Why? Before they can start, there must be an existing 3' end. So they can, first thing is the DNA polymerase can only add. They cannot begin. You want to make that in your notes. DNA polymerase can only add. They cannot begin. That's number one. Number two, even when they are going to add, they must be adding to an existing surface. That existing surface must be 3'. So for them to add, they had to add to this existing surface. I'll talk about that later. So because they have to add, something has to begin because they can only add. So they cannot add to 0. So something must begin for them to add. So RNA must be made first. And so an enzyme will come and make RNA. That enzyme is called primase. So it's going to make RNA strands. So this red is RNAs made by primase. I'll take it again. When two DNA strands are twisted together, topoisomerase need to go first to relieve the pressure of the twist. Number 2, helicase then go and untwist them. As helicase untwists them, an enzyme, single-stranded binding protein, single-stranded binding protein is going to bind on it to prevent them from recoiling on each other. Replication fork has been opened. Should we start making DNA? Yes, but we cannot make DNA. Why? Because the enzyme that makes DNA, DNA polymerase, cannot begin the process. It can only add to the process. So to begin the process, we have to use an enzyme that can begin, and that is RNA primase. Primase can create RNA first, but nothing can create DNA first. So we'll now make an RNA first. All right, so I'll continue from there. So synthesizing a new strand, DNA polymerase require a primer to which they can add. Can you see that? They need to add. DNA polymerase can only add, they don't begin. So the initial nucleotide chain is a short RNA primer that is created by primase. So before DNA is made, RNA has to be made first. The completed primer is 5 to 10 nucleotides long, about that. The new DNA strand will start from the 3' end of the RNA primer. So if DNA polymerase does not see 3' end, which is this, if DNA polymerase does not see this, DNA polymerase cannot start. It can only add to that direction. DNA polymerase cannot add to 5', it can only add to 3'. So if the 3' does not exist, DNA polymerase cannot do any job. Let's go. So enzyme cupped DNA polymerases catalyze the synthesis of new DNA at a replication fork. Most DNA polymerase require a primer, you see that, and a DNA template strand. So the DNA template strand is a strand from the parent. The primer is an RNA. The rate of elongation is about 500 nucleotides per second in bacteria and 50 per second in human cells, of course. Each nucleotide that will be used, like each building block of DNA, will be added to a growing strand that is the 3' strand. So each nucleotide that is added to a growing DNA strand, is a nucleoside triphosphate. Deoxyribonucleotide, ATP, supplies adenine to DNA and is similar to the ATP in energy metabolism. The only thing is that the one is deoxy. I'll talk about that later. The difference in their sugar is that this sugar has the deoxyribose sugar, while the ATP has ribose sugar. ATP have more orientation like an RNA, but DNA have deoxyribose sugar, ATP have ribose sugar. Now, what is deoxyribose sugar? So as each monomer joins the DNA strand via a dehydration reaction, it loses two phosphate groups as a molecule of pyrophosphate. I'll talk about that. Give me a moment. Let me check my computer. Looks like the battery is okay. So you see the template strand. This is going to serve as a template strand, which is coming from the parent, of course. This is the template strand, which is the parent strand. And then the new strand is going to be this. And again, you see this parent template strand is on 3' to 5', and new strand is 5' to 3'. So it's alternated on each other. So that's the way the DNA polymerase works. And I will show you more of that as we go on. All right. So antiparallel elongation, the antiparallel structure of the DNA is going to affect the fact that one is going to be in 5' to 3', another is going to be in 3' to 5'. That's going to affect how the DNA is going to replicate, because think about it, one will be made in 3' to 5', the other will be made in 5' to 3'. And I told you that DNA polymerase can only add on one side, on the 3 side. So you're going to see what I want to show you now. So the antiparallel structure of the double helix is going to affect the replication. DNA polymerases, like I said, DNA polymerases are only able to add only to the free 3' end. DNA polymerase cannot add to 5' end. They can only add to 3' end. Now, therefore, a new DNA strand can elongate only in the 5' to 3' direction. So the only way it can happen is if the DNA polymerase is adding nucleotide to 3' direction. So look at what is going to happen now. So along one template strand of DNA, the DNA polymerase synthesize what we call leading strand, and the other we call it lagging strand. Leading strand means it's continuous. The other strand means it is discontinuous. And I'm going to show it to you very simple here now. So when the replication fork opens and the DNA strand is being secreted, is being synthesized, what is going to happen next is that you see this is the origin of replication fork open, the primer is secreted here. Now at this point, the template strand is the dark. This is the template strand, right? This is the template strand for the other one. So the template strand is starting from 3' to 5'. That means the new strand that will be made on this inside here is going to be 5' to 3'. So it can only be made in this direction. You can only add nucleotide this way because you can't hide it here. But the other strand now, which is 5' to 3', is going to also have its new DNA as 5' to 3'. So that means the new DNA is going to be 5' here, 5' here, and 3' here. So that means it is easy to add nucleotide to this 3' end because the 3' end is open. So as the DNA replication, DNA opens, so it makes the strand. So the strand that is being made that is okay. Now, this I will show you that here. The strand that cannot be made in the continuous manner, we call it lagging strand. The strand that can be made in the continuous manner, we call it leading strand. So if you look at this now, for instance, when the strand here is opened, think about this with me. When this is open, an RNA primer is made here. Don't forget the parent strand is 3' to 5'. So the new strand we want to make has to be 5' to 3'. So immediately that happens, you see DNA polymerase can come in, DNA polymerase 3, right, come in and start to make the DNA strand, because 3' end is open here. So it starts to make the DNA strand. And as this fork open, it continues to make it, continues to make it straight away. So this strand is going to be our leading strand because it's continuous and it's easy to make. So you see, this is just all that happens. How many primer is needed? Just one primer. And as the fork opens, you see topoisomerase helicase opens it, and it keeps opening, it keeps making open, and the DNA polymerase will keep making the new DNA. So this is what we call leading strand or continuous strand. Now let me show you the other challenge that we are going to have to overcome. Now, there is another strand that cannot happen in that direction, and we call it lagging strand. The lagging strand has to be synthesized in fragments, small fragments, and we call that Okazaki fragments. Then the fragment will now later be joined together by what we call an enzyme that is called DNA ligase. So look at it. So I already showed you how the leading strand or continuous strand is made. But the other one, we call it discontinuous strand or lagging strand. So if you look at this now, the parent strand here is 5' to 3'. So that means the new strand is going to be 5' here to 3' here. So that means when the replication fork is opening, we cannot make DNA strand, we cannot add it to this 5' end. No. We cannot add it to this 5' end. No. So what we have to do is to make multiple primers. So the first thing is this: we make this primer. After making this primer, then we can add DNA strand to this side. So we make a new DNA strand here. Then we make another primer here. Let me use red for primer. We make another primer here. And then make new DNA strand to it. So see, the challenge is because the orientation of this strand does not allow us to make one primer and have open 3' end. The open 3' end is always going to be on this side. And if it is on this side, we cannot add it this way. We have to add it this way. So multiple primers will need to be made. So you see, as this is made, another primer is made. And another primer will be made here, then it will be made again. And that keeps going because you cannot make one primer for this case. You have to make multiple primer and make multiple Okazaki fragments. So this is called Okazaki fragment. Number 2 is another Okazaki fragment. And now at the end of the day, DNA polymerase 1, DNA polymerase 3 was the one doing the job before. You see that? So DNA polymerase 1 is now going to come in because it's smaller. 1 is smaller than 3, right? 3 cannot come in between. 3 can only work on the stereo. So we call it exonuclease activity. So we say 3 have exonuclease. I'm going to hide this here. DNA polymerase 3 have exonuclease activity. It's only able to work where there is external space. It cannot go in between. But DNA polymerase 1 have endonuclease activity. So it's able to go in between. Let's say it's because it's small, right? So that's that. So DNA polymerase 1 will now come here now, come to this middle and cut out this. You see, it has cut out this. And then another DNA, so it will cut out this and replace it with actual DNA segment. So it will put DNA segment there. So you see DNA is here, but now there's still a little gap here. Now ligase is also an enzyme. DNA ligase is going to come and fill up that space, which is the little gap that is remaining. Does that make sense? All right. So that is how DNA replication happen in the lagging strand. All right. So again, this is what we have eventually. We have now the lagging and the leading strand. So you see, the leading strand can be made straight up, just one. So look at this here. When the leading strand can be made straight, one primer, and it will just be made straight. But the lagging strand cannot be made like that because DNA can only be added, DNA polymerase 3 can only add in the 3' direction. So we have to make the primer this way, make the Okazaki fragment, make another primer in front, make Okazaki fragment, make another primer in front, make Okazaki fragment, make another primer in front, make Okazaki fragment. Then later, DNA polymerase 1 is going to go cut out the RNA primer because we don't want to have an hybrid DNA and RNA together, right? So you have to cut out the RNA primer. Then DNA polymerase 1 does that and fill it with nucleotide so that it will now have DNA there. But little gap that is remaining there, DNA ligase go and fill up that gap so that it will be connected. So that is all that happened with DNA replication. Major thing you want to take note is take note of the function of the enzymes, which I've talked about. That is the key thing. This is just showing you the thing again. Now, proofreading and repair. DNA polymerases proofread newly made DNA, replacing any incorrect. So as DNA polymerase 3 is synthesizing the new DNA, it also have the ability to read. Now, it is reading it as it goes. If any mistake happen as it's reading it, it's able to fix the error. But if a mistake, if a mistake happens and it's already past what it's doing, it cannot go back in between. You know, I told you that DNA polymerase 3 does not have endonuclease activity, it only has exonuclease. So DNA polymerase 3 or DNA polymerases proofread newly made DNA, they read it as they go. In mismatch repair, mismatch means that they have put in a wrong nucleotide, a wrong nitrogenous base where they should be, right? Repair enzyme replace incorrectly paired nucleotide that have evaded the proofreading process. So I'm going to show that to you now. All right, so this is what we call DNA repair. Now, DNA can be damaged by exposure to harmful chemicals, sunlight, all of that can cause DNA damage to DNA. So when there is damage, what is going to happen? DNA polymerase is going to go, nuclease is going to go, cut that off, then DNA polymerase is going to fix that and put, of course, this is going to be DNA polymerase 1, not DNA polymerase 3. And then ligase go and fill up the gap that is there. It's as simple as that. Now, evolutionary significance of DNA altered nucleotide. All right, of altered DNA nucleotide. The error rate after proofreading and repair is low, but it's not zero. All right, sequence changes may become permanent and can be passed on to next generation. These changes, we call it mutation, are the reasons why we have variations in genetic composition and expressions, and of course, diversities. Now, for linear DNA, the usual replication machinery cannot complete the 5' end of daughter DNA strand. There's no 3' end of a pre-existing nucleotide for DNA polymerase to add on, right? Thus, repeated runs of replication produce shorter DNA over time. And so it ends up happening that the DNA will become shortening over time. This is not a problem in prokaryotic, most of which, of course, because they are circular DNA. All right, so this is what we are talking about. So because the DNA does not start by itself, right? So the last end of the DNA is usually not going to be synthesized, right? So it's usually like lagging. So gradually, DNA becomes shorter over time as replication continues. So eukaryotic chromosomal DNA molecules have special nucleotide sequence at their ends, which we call telomeres. Telomeres do not prevent shortening of DNA molecules, but they do postpone the erosion of genes near the end of the DNA. Does that make sense? They don't prevent it from shortening, but they prevent help to preserve the genes in that end. It has been proposed that the shortening of the telomeres is connected to aging, of course. If chromosome of germ cell became shorter in every cell cycle, essential genes would eventually be missing from the gametes they produce. An enzyme called telomerase catalyzes the lengthening of the telomeres in germ cells, which will help to prevent losing of those genes. The shortening of the telomeres might protect cells from cancerous growth by limiting the number of cell divisions. There's evidence of telomerase activities in cancer cells, which may allow cancer cells to persist. All right, so the bacteria chromosome is double-stranded as well, but it's circular molecular DNA molecule associated with a small amount of protein. Eukaryotic have linear DNA, right? And it has large amount of protein. In bacterium, the DNA is supercoiled, found in the region of the cell called nucleoid. In eukaryotic, the DNA are linear and they are located in the nucleus. All right? They are located in the nucleus. When they are condensed, we call them chromosome. When they are not condensed, we call them chromatin. Chromosome fit into the nucleus through an elaborate multi-level system of packing. The nucleus, the DNA is packed around by histone protein. So protein called histones are responsible for the main level of DNA packaging. All right, I've talked about that. Okay, so the unfolded chromatin resembles beads in a string. Each bead is being called nucleosome. So we call it nucleosome. But however, we know that. So DNA and histone protein makes what we call nucleosome. The nucleosome, when it's condensed chromosome, when it is uncondensed chromatin. A nucleosome is composed of DNA one twice around by a core of 8 histone protein. You see that? Now, the main end of each histone protein extends outward from nucleosome and is involved in regulation of gene expression. All right? So I'm going to stop there. I've talked about chromatin, uncondensed chromosome. All right? So I'll stop there. If you have questions, please reach out to me. Have a great time.